Module 4, Part 3. So in this part, we will begin discussing the third type of cytoskeletal filament, and that is microtubule. It is made up of tubulin monomers. You will remember that I told you that these are the largest of the three filaments, about 24 to 25 nanometer in diameter. They are hollow or cylindrical in shape and very rigid as compared to the other filaments, actin and intermediate filaments. And I also mentioned that you usually see microtubules as long, straight structures in the cell, emanating from what are known as microtubule organizing centers, as shown here, right here. I will elaborate on these tubules in this part. So I want to make the point that these are quite large. You can see them on electron microscopy, and this is a transmission EM of the microtubules, and you can clearly see the cylindrical structure of these filaments. So microtubules play a number of important functions in the cell. They are responsible for organizing a lot of intracellular organelles and serving as roads for transport of vesicles. Of course, the transport of vesicles on these microtubule road is carried out by microtubule-associated proteins, which are the motor proteins, which we'll cover later part of this topic. Then they are important in beating of cilia and flagella on certain cells, and they provide structural integrity to nerve cell, red blood cells, and also platelets and to the flagellar structures. And finally, they are responsible for the alignment and separation of chromosomes during different kinds of cell divisions, such as mitosis and meiosis. In this part, we are going to cover the first three. Then later on, when we talk about cell division, we will revisit the microtubules and see how they function in helping to separate the chromosomes and distributing chromosomes to the daughter cells during the cell division. As I mentioned, microtubules are made up of monomers of tubulin, and tubulin itself is a heterodimer. So it has two subunits, and the two subunits are alpha tubulin and beta tubulin, shown here in the light green, and the beta tubulin shown here in the dark green. These tubulin dimers, this heterodimer, interact with each other from end to end to form a protofilament, which is shown here. One of them is outlined here, right here in the yellow. And these protofilaments interact laterally to form this tubular structure. You will recall that when we talked about the actin and actin polymerization, we saw that the actin monomers could bind ATP. In contrast, the tubulin monomers can also bind nucleotide, but in this case, it is GTP. And each tubulin heterodimer can bind two molecules of GTP. So one GTP binding site is on the alpha tubulin, and one GTP binding site is on the beta tubulin. The site on the beta tubulin is known as the exchangeable GTP binding site because it is this site where you get GTP binding, GTP hydrolysis to GDP, and the exchange of GDP for GTP. And the GTP on the beta tubulin monomer that influences polymerization and depolymerization of the microtubules. On the other hand, the GTP bound to the alpha tubulin monomer is permanent. It is very stable, and it is thought to have structural role there because it is never hydrolyzed to GDP or exchange. There is a third kind of tubulin that is known as the gamma tubulin. It is drawn here in this circle and plays an important role in nucleating the formation of the microtubules. You may also find gamma tubule in microtubule organization center, but gamma tubulin is never the part of the final microtubule structure. Most of these microtubule in the cell are in these singlet tubules, and they are usually made up of 13 protofilaments. But in certain genera, you see the protofilaments that can itself be as small as 11 protofilaments or as big as 15 protofilaments, but these are extremely rare. Microtubule can also be found as doublets, as shown here. You usually see the doublet microtubule in cilia and flagella. If it is doublet, then the protofilament are 13 in the A ring and then 10 in the B ring. You can also observe triplet microtubules within the cell and usually see them within the centrioles with 13 plus 10 plus 10 arrangement of the protofilaments. In centrosome, which are important microtubule organization centers, and in basal bodies within the flagella and cilia, which are also important microtubule organization centers. We are going to elaborate on these doublets and triplets and their function in the cell in a little bit. There are two main populations of microtubules in cells. There are unstable short-lived microtubules, which assemble and disassemble rapidly according to the needs of the cell. So these are microtubules that must shrink and grow when cells divide and when tubules are needed for the movement of organelles or vesicles around the cell. So they are very unstable and very dynamic. So here is an example. Here are microtubules in this larger picture. This is an immunofluorescence image. The microtubules are stained in green. You can clearly see them emanating from the microtubule organization center. This would be a cell in the interface, so it is not dividing, and this would be a typical arrangement of microtubules in an animal cell, in a mammalian cell. But when this cell gets ready to divide, all of these microtubules will depolymerize, and the tubulin monomers are used to building blocks to form the mitotic apparatus as shown here in the inset. So they have disassembled and reassembled into this structure, which is real important in segregating chromosome, and we'll come back to this later. So that's an example of unstable short-lived microtubules. But there are stable and long-lived microtubules that remain polymerized for a long time. And in some cells, especially in non-living cells, they remain polymerized forever. You can see these kinds of microtubules which never change their length, they never shrink or grow. You can see them in the flagella of sperm cells. You can see them right below the plasma membrane of the red blood cells, which is known as the marginal band region. You can see them in platelets, and you can see them in the axon and the dendrites of the nerve cells. And what these long-lived stable microtubules do is that they provide structural integrity for the cell. And this makes sense. Some axons on nerve cells are extremely long, and so you can imagine that they would be extremely fragile. But if you look at the EM of an axon from a nerve cell, you can see it is just packed with microtubules and intermediate filaments, and this provides structural integrity to the cellular outgrowth. In the case of marginal band, you can imagine that red blood cells are squeezing through capillaries. There is a strong possibility that they could be damaged because capillaries are so small, but this population of stable microtubules just below the plasma membrane provides structural integrity and protection of red blood cells when they are moving through the capillaries. So I mentioned a couple of times microtubule organization centers, so let's elaborate on that. This is the structure used by a cell to nucleate and organize microtubules. The most common one that you will see is the primary microtubule organizing center in the animal cell, which is known as the centrosome. So in this immunofluorescence image, the microtubules are stained in green, and some components of the centrosome is stained in yellow fluorescence. You can see the microtubules are emanating in a spoke-like fashion from the microtubule organizing center, which is the centrosome in this case. So let's take a look at some diagrams of microtubule organizing center. So here is a centrosome. This is an interface animal cell, and this particular animal cell happens to have a flagella or a cilium. This pink region is known as the centrosome, and inside the centrosome are short triplet microtubules known as centrioles. Together, this makes up the microtubule organizing center, and you can see these microtubules emanating from this structure. The minus end of the microtubule is interacting with the centrosome, and the plus end of the microtubule is near the plasma membrane. I should mention at this point, but we will cover this a little later. Like actin, the two ends of microtubules are not biochemically equivalent. So there is a minus end and a plus end, just like in actin. Then you will find microtubules in the flagellum or cilium in the cell. That is actually these structures. At the base of the cilium or flagellum, which is known as a basal body, which contains the triplet microtubule. And this is another type of microtubule organizing center. And again, you can see that the minus end is interacting with this microtubule organizing center, and the plus end is towards the periphery of the cell. So here is the orientation of the cellular microtubule in a dividing mitotic animal cell. In this case, you have two microtubule organizing centers. They are known as the spindle poles, and they arise by the division of the centrioles. When a cell is in the interface, there is a single microtubule organizing center, and it is a centrosome with centrioles. The centrioles can divide, and then you can get two microtubule organizing centers in the mitotic animal cell. And again, the minus ends of the microtubules are interacting with the organizing center. The plus ends are the farthest away from the microtubule organizing center, and the role of these microtubule is to segregate the chromosome during the cell division. So we have already talked about the nerve cell, but here is an organization of microtubules in a typical nerve cell. The axons are responsible for sending signals away from the nerve cell body to be received by another nerve cell. They might be situated right here at the end of the axon. When you have dendrites, and dendrites are these branches or outgrowths of the nerve cell, and it is through the dendrite that these nerve cells receive the signal. So the axons for sending the signals and the dendrites are for receiving signals. You have a typical organization of microtubules in the cell body where you have the organizing center that contains the centriole and the microtubules emanating from the organizing center. And then you have these very long-lived microtubules in the axons and the dendrites. Now, the first thing that you should notice about this, that there is no microtubule organizing center in the axons or the dendrites. So this is the one example where there is no microtubule organizing center. These microtubules that are in the axons or the dendrites of the nerve cells are there, that they are not organized by some kind of organizing center that contains triplet microtubules within it. The other thing you will notice is that the orientation is not uniform. So in this axon, you notice that these microtubules that are varying in the length, some short ones, some long ones. The minus end are all towards the nerve cell body. The plus ends are pointed towards the extreme end of the axon. In the case of dendrite, you have this long microtubule within the structure, but they are not all at the same orientation. You have some with the plus end towards the nerve cell body and some with the minus end towards the nerve cell body. Again, in this case, they are long-lived stable microtubules that provide the structural integrity of the cell. If you understand the polymerization and depolymerization of actin, then you already understand polymerization and depolymerization of tubulin because all of the features are the same. The two ends are not biochemically equivalent. There is a plus end, which is a fast-growing end, and there is a minus end, which is a slow-growing end. In vivo, you almost always get addition at the plus end and release of subunit at the minus end. This is because of the critical concentration. There's a lower critical concentration for the plus end than the minus end, so you are more likely to get growth at this end and loss at the minus end. And because of the unequal growth and loss at the end, you can also observe the phenomenon known as the treadmilling, which you saw in the actin. So again, if you understand what is going on in actin, all of the same features apply for the tubulin and the microtubules as well. So you will notice that when we were discussing about microtubules, we mentioned that the minus end was always associated with the microtubule organizing center, and the plus end was the farthest away from the organizing center. How do you know that? So now that you understand the polymerization of tubulin, we can look at an experiment that demonstrated that the minus end is what is associated with the microtubule organizing center. If you take an interface cell, here is your center of the microtubules, and here is the organizing center. You can cool those cells to 0 degrees, or you add a drug known as coulcimate. The microtubules will depolymerize. Eventually, what will happen that all of those microtubules in the cell will depolymerize as shown here. Here is your existing microtubules organizing center. However, if you wash out the coulcimide, it happens to be reversible. Or if you warm the cell to 37 degrees, which is the normal growth temperature, you begin to see the growth of the microtubules out from the microtubule organizing center. So the microtubule organizing center will nucleate, not only organize the microtubules, but will nucleate the formation of new microtubules. Since we know that growth is happening at the plus end and it grows from the microtubule organizing center out, then you can make the conclusion that the minus end must be associated with the microtubule organizing center and the plus end must be farthest away. So this is an experiment that shows that the minus end are associated with the microtubule organizing center. So here is just a comparison of microfilaments or actin with microtubules just to take home the point, the amazing similarities between these two filaments. They are both polar, they have plus ends and the minus ends, which are not equivalent biochemically. They both bind nucleotide, and in this case of actin, it is ATP, and in the case of microtubules, it is GTP. What subunits can polymerize? In vivo, it is the actin ATP that polymerizes, or tubulin GTP. In vitro, if you raise the concentration high enough, you can force the actin ADP to polymerize, tubulin GDP to polymerize. And also in vitro, you can use a non-hydrolyzable analog of ATP, and those can polymerize in vitro. And likewise, you can take non-hydrolyzable analogs of GTP analogs. We talked about this experiment when we talked about actin. What is required for depolymerization? You must have ATP hydrolysis before you can get depolymerization. So you must have a conversion of ATP to ADP before actin will depolymerize. And likewise, you must have a hydrolysis of GTP to GDP on the beta tubulin before the microtubule will depolymerize. Of course, it is the use of these non-hydrolyzable analog that led to these conclusions about the depolymerization. So similar to actin, there are some interesting drugs or small molecules that can be used to study microtubules. One is known as a colchicine, which binds between the alpha and the beta tubulin on the dimer. At very high concentration of colchicine will depolymerize tubulin. At a very low concentration, it will stabilize the microtubules. In either case, it is detrimental to cells. If their microtubules are depolymerized or if their microtubules are artificially stabilized, this is very detrimental, especially to the cell division. Another drug that you can use is known as Taxol. This can bind to the side of the microtubule to stabilize them. You can use these drugs to study the function of microtubules within the cell. For example, if you are interested if the microtubules play a role in phagocytosis, you could treat the cells with high concentration of colchicine such that all the microtubules will depolymerize, and then you can feed these cells some type of phagocytic target. Then you can ask the question, can they still phagocytose the target even in the absence of microtubules? And in doing so, you can answer the question whether the microtubules are necessary for the cellular function. So you may have heard about colchicine and Taxol before. They are widely used as chemotherapeutic agents. They are often used to treat cancer patients. One of the main functions of microtubule is cell division, which we're not talking about here, but we will talk about later when we talk about the cell cycle. But you can imagine if you disrupt the microtubule function, you will severely inhibit cell division. This is what you want to do in the case of a tumor cell. Tumor cells are the cells that lost the ability to control their cell division. They are dividing rapidly, and it is completely unregulated growth, and that is why they form the tumors. By exposing tumor cells to the colchicine or Taxol, you can inhibit its cell division, and this will stop the growth of tumor cells. They are very potent and widely used chemotherapeutic agents, but they are also widely used in the cell biology lab, and we ask specific questions about the role of microtubules in other cellular function using these drugs. Now, in this section, we are going to concentrate on proteins that associate with the microtubules. We will cover 4 main classes of proteins. We will start with the microtubule associated proteins or MAPs. This group of proteins will confer stability to microtubule structure, especially those long-lived or permanent microtubule structure that you see in a neuronal cell and in the flagella of motile cell. Then we will look at the proteins that regulate polymerization at the plus end, and this same class of proteins can also connect microtubules to other structures. This class of proteins are known as TIPS, which stands for the plus end tracking proteins. Then we are going to look at proteins that regulate disassembly at the plus end, which is necessary during cell division. Finally, we will look at a class of proteins known as motor proteins, and you will remember that motor proteins are those that use the energy of ATP hydrolysis to move along the filament. When we were talking about the motor proteins, we will also consider intraflagellar transport and genetic disorders of flagella known as ciliopathies. So let's start with the first group of proteins, that microtubule-associated proteins, or MAPs. The best characterized proteins are MAP2, MAP4, and tau. MAP4 is not shown on this slide, that only MAP2 and the tau. This group of proteins all have the same basic structure in that they have two domains. One domain is made up of repeats of positively charged amino acids, shown here. This part of the protein interacts with the negative charge that you see on the surface of the microtubules. The other domain of the protein sticks out from the domain interacting with the microtubule at a right angle, and this serves as a spacer to stabilize the microtubule structures. So here you can get a good idea how these types of proteins can stabilize and space microtubules. Here's a microtubule bound to the MAP2 that have much longer spacer arm than the tau, and you can see that the microtubules that are bound by tau are much closer together. So it's pretty clear that how MAP can space and organize the microtubule in the cells. So the different functions of the MAPs and the taus. So in the second group of protein that regulate polymerization at the plus end, and some examples are EB1 or EB3, which stands for end binding proteins. We mentioned that in vivo, you almost always get addition at the plus end and loss or disassembly at the minus end. And this is due to the differences in the critical concentration at each of these non-equivalent biochemical ends. There are also some proteins that can enhance the polymerization at the plus end that can add to the growth, and you often see that at the plus end. These proteins are plus tip proteins. In fact, they do a couple of things. First of all, they can promote growth at the plus end. They can also prevent disassembly at the plus end. So they stabilize the plus end, and they can also connect the plus end of the microtubules to other structure in the cell like actin or membranes. So here is an immunofluorescence image. In this image, the microtubules are stained with a red fluorescence fluorophore, and EB1 plus tip protein is stained in green. You can very clearly see the EB1 at the end of many of these microtubule filaments, and these are the plus end because these end-binding proteins have an affinity for the plus end. So here is a higher magnification of these end-binding proteins interacting with the plus end microtubule. So here is a very interesting diagram which is known as a chymograph. A chymograph shows the dynamic instability of microtubules. In other words, in chymographs, one can discern what part of the microtubule are growing and what parts are shrinking. These solid arrows are areas of microtubule growth, and these reverse dotted areas are the areas where microtubule is disassembling. In green is the EB3 staining. You can very clearly see that EB3 is associated with the areas of growth, and in the areas of disassembly are pretty much devoid of EB3. This really shows you that these end-binding proteins associate with the plus end of the microtubules, and in particularly growing end of the microtubules. The areas labeled as C are called the catastrophe, where there is an abrupt shrinkage from the growth occurs, and the labeled R regions are called the rescue, where the growth resumes from the shrinkage. So this bottom right diagram, on the other hand, depicts the current view of the end-binding proteins, how they interact with the microtubules. It is thought that there is a special structure at the plus end that attracts the end-binding proteins to that particular portion of the filament, but further research is needed in these areas to prove this hypothesis. When we think about microtubules, we usually think of just growth at the plus end and disassembly at the minus end, but there are occasions, especially during the cell cycle, where you require depolymerization at the plus end. This is mediated by a number of proteins, and the best characterized of these are known as kinesin 13 and strathmin. And I will show you in a minute that when you need depolymerization at the plus end. Kinesin 13 is interesting because it actually is a part of the microtubule motor protein known as kinesins. These proteins are the plus end directed motor, which means they move along the microtubule filament from the minus end to the plus end. Kinesin 13 is put in this family of these motor proteins because of the homology to these proteins. It shows amino acid and structural homology to the kinesin family, but it doesn't behave like a typical motor protein. In fact, the kinesin 13 is that what interacts with the tubulin subunit at the plus end of the microtubule, and it causes the microtubule at the plus end to bend as shown here. And when that microtubule bends, it destabilizes the microtubule, and this promotes the disassembly or depolymerization at the plus end. The other protein, the strathmin, interacts with the plus end or near the plus end of the microtubule, but what it does is that it enhances the GTP hydrolysis by the tubulin dimers. Remember that tubulin dimers, which are building blocks of the microtubule, can bind GTP, and when they get incorporated into the microtubule, they are GTP bound. But then as they reside in the microtubule filament, the GTP is hydrolyzed to GDP, and this destabilizes the structure. So what strathmin does that it enhances or speeds up this GTP hydrolysis, and in doing so, it destabilizes the portion of the microtubule faster than it would normally be destabilized. So what we are seeing in the depolymerization at the plus end, probably the best characterized occasion of the plus end disassembly is during mitosis. We saw this diagram already. This is a diagram of a mitotic animal cell showing the organization of microtubules emanating from the two spindle poles, which are microtubule organizing centers. You can see that each of the daughter chromosome is interacting with the plus ends of these microtubules. And here are the minus ends interacting with the microtubule organization center. When it comes for the cell to divide, or when it comes time for these daughter chromosomes to be delivered at each side of the dividing cell, then you get rapid shrinking of the microtubule towards the microtubule organizing center, the spindle poles. And that occurs in part by depolymerization of the plus end. It is the proteins that I just mentioned, kinesin 13 and strathmin, as well as some others, that can promote disassembly at the plus end, even though it is not biochemically favorable. In vitro, you most always get addition at the plus end and loss at the minus end, but this is true for most occasions in vivo as well. But there are some exceptions like this. So let's move on to the fourth group. These are microtubule-based motor proteins. And you will remember that we talked about motor proteins that associate with actin. We defined a motor protein as one of that uses the energy of ATP hydrolysis to move along the filament. In the case of actin, the family of motor proteins is known as myosin, with the exception of myosin 6. All myosin motor proteins move towards the plus end. Well, in the case of microtubules, you have two major classes of motor proteins, the plus end directed motor and the minus end directed motors. Plus end directed motors are known as the kinesins, whereas the minus end directed motors are known as the dyneins as they move towards the minus end. Here is a table that takes home the point that this is a huge family of proteins. You have microtubule motor proteins that function in the cytosol. You have specific motors that function during mitosis. There are specific motors that function in the exonym, which is a part of the flagellum or cilium. So we are going to look in general the cytosolic kinesin, the cytosolic dyneins, and the axonal dyneins. So here is a general structure of a cytosolic kinesin, very much look like an actin-based motor myosin. So it is made up of two heavy chains. They are globular heads, which is the site of the ATP hydrolysis and the site of interaction with the microtubules. You have this tail region where the tails of the two heavy chains interact in a coil alpha helix manner. And then you have several light chains that are bound over here. Those light chains that mediate the interaction between the motor protein and its cargo, such as a vesicle or an organelle or a membrane. So there are a couple of examples of cytosolic kinesins. You have kinesin 1, which is the conventional kinesin that has two heavy chains and several light chains, as I just mentioned. There is kinesin 2. The structure looks very similar. What distinguishes it from the conventional kinesin is that it is heterotrimeric, in that it has two different heavy chains and a single light chain. So it is made up of three different proteins. That is why it is called a heterotrimeric kinesin. The role of kinesin 1 and kinesin 2 are to bind to vesicles and organelles and to walk along the microtubules towards the plus end to traffic vesicles in the cell and to organize the organelles within the cell. Kinesin 5, it is interesting because it is made up of a number of heavy chains. When the tails interact, you end up with two globular heads on one side and two globular heads on the other side. And because of that, the kinesin 5 can interact with two different microtubules simultaneously. This kinesin can actually organize microtubules relative to one another, or they can move microtubules relative to the other microtubules. Kinesin 13, as I mentioned, is a part of the kinesin family because of its homology, and it's the function to destabilize the plus end, and it also does the same thing at the minus end. So it binds to the end of the microtubule and bends them to destabilize them, and we already talked about this one. So here is a very simple diagram that how a cytosolic kinesin moves a vesicle. In the next slide, we will work at this in more detail. Here is a cytosolic kinesin with two globular heads and is interacting with the microtubule. The light chain at this end is interacting with the vesicle, and through the hydrolysis of ATP, these kinesin walks along the microtubule to the plus end in a manner that is very similar to the way the myosins move along the actin. So here is what it looks like in greater detail. Here are the two heads that are bound to the microtubule. The current leading head is not bound to any nucleotide. And when the globular head is not bound to the nucleotide, it interacts with the microtubule very tightly. And remember that this is true for myosin as well. When the myosin molecule is bound to an actin subunit and it is not bound to a nucleotide or ATP, this interaction is very strong. In step 1, what is happening? That you get a binding of ATP by this subunit, and this causes it to loosen its interaction with the side of the microtubule. In the step 2, the head that was lagging or behind swings forward, and it is now the leading head. And it is now in the front. It stepped forward. In step 3, the leading head binds to the microtubule, and in the step 4, the leading head releases the ADP, and since it is not bound to the nucleotide, the interaction of the head and the microtubule is very light. And at the same time, the lagging head hydrolyzes the ATP to the ADP, and the cycle again continues or repeats again. So this is a very similar way that the myosins walk along the actin. So that was a cytosolic kinesin, and that is all we are going to say about them. So now let's take a look at the cytosolic dyneins, which is the other microtubule-associated motor protein that walks towards the minus end of the microtubule. These are enormous protein with multiple subunits. We're not going to talk about all the different subunits in much detail and all the different functions of these subunits. We are just going to look generally to the structure and generally how it functions. So the dynein contains two large, greater than 500 kilodalton, two intermediate, and two small subunits. It has two heads similar to the kinesin. The dynein heavy chain domain has a stem, then you have a linker, and then you have a 6 AAA ATPase repeats number 1 through 6. AAA stands for ATPase associated with diverse cellular activities, which is a superfamily of proteins with conserved 231 amino acids. Then you have a stalk and its microtubule binding domain between the AA repeats 4 and 5, and an alpha helical domain that supports the stalk. The 6 AAA repeats form a circular structure with a coil-coil stalk domain that contain the microtubule binding domain at one end. The dimerization and the intermediate chains associated with the stem region that can link the dynein cargo with the dynactic protein complex that we are going to see. So this is the dynactic binding area. So the 6 AAA repeats form a circular structure with a coil-coil stalk domain containing the microtubule binding domain at one end, the dimerization domain, and an intermediate chain, and associated with the stem region that can link the dynactin protein complex, which binds towards the cargo end. So the dynactin is a very complex multi-protein complex, and dynactin links dynein to the cargo and regulates dynein activity. At the top, you can see that there is one of the two dynactin domains. It has a short filament of one actin subunit and 8 actin-related proteins, and ARP1 subunit capped by cap Z at the equivalent of the plus end in the actin. This cargo binding domain subunits of the first domain connect the dynactin to the cargo through an adapter. The second subunit is responsible for the dynactin, dynein, and the microtubule interaction. In the second domain, the P150 glued protein attaches the dynactin to the dynein and to the microtubule. Dynamitin subunit holds these whole complex together. And interestingly, the reason it was named as a dynamitin, because if you overexpress this protein, instead of associating the dynactin complex with the microtubules, it blows this whole thing apart. And the dynactin P150 glue subunit binds to the EB1 to associate dynein with the growing plus end of the microtubule. And quite simply, this is how it works. In this case, we are just looking at one half of the cytosolic dynein. So you have the microtubule binding domain interacting with the microtubule. Here is the ATP head domain, and here is the other side of the stem that is interacting with some kind of cargo that is bound, possibly a membrane, through a dynactin complex. What happens is that upon ATP hydrolysis is what you get a rotation of the head region and change in position of the head region with the microtubule binding domain. In this particular example, if the dynein is associated with some kind of a permanent structure, such as a membrane, then this rotation will call is to slide the membrane from the direction from the right towards the left. So this was the initial position, and it moves towards this position. It moves from here to here. Remember that also, that dynein always moves from the plus end towards the minus end. So it is going towards this direction. What is interesting is that you can see why the cell needs both plus end and the minus end directed microtubule motors. So you can see on this diagram all of the microtubule emanating from the microtubule organizing center right here. And you can see the endosomes and the lysosomes right here. And you can see that the endosomes and the lysosomes, which are associated with the cytoplasmic dyneins, moving towards the center of the cell. You can see the secretory vesicles that are associated with kinesin moving towards the plus end, which is the periphery of the cells. And there are even some organelles that are associated with both motor proteins. For example, the mitochondria is associated with both a kinesin and a dynein, and it can move in either direction along with this microtubule. So you can see that the cells can regulate the vesicle trafficking, and it can organize all of the organelles within the cell. The kinesin can interact with a portion of the ER in the ER membrane extension and move them towards the periphery of the cell. And this is how you can get the lacy network pattern that you can see with the endoplasmic reticulum. It is because of the association of the motors and how these motors connect and move that ER along the microtubule. That gives you that kind of arrangement. You can also have cooperation in between actin-based motor proteins and the microtubule-based motor proteins. So here is another of a vesicle that is moving toward the plus end of the microtubule with the help of a kinesin. But let's say the microtubule doesn't reach the cell periphery, and yet this vesicle needs to be at the edge of the cell. This vesicle can become associated with the myosin and then can jump from the microtubule to the actin microfilament and continues its journey towards the outside of the cell. So you can get cooperation between the kinesin and dyneins, and cooperation between microtubule-based motors and also actin-based motors. So now let's take a look at the exonymal dynein. There are some motile cells use a structure such as a flagellum or cilium to move. So they require or rely in this organelle for locomotion. The structure of flagella and cilia are the same. The main difference is how many there are per cells. So for example, cells that have a flagella only have 1 or 2 flagella per cell. And again, they are used in locomotion. There are a couple of single cell eukaryotes. And of course, the sperm cell use the flagella to move. For cilia, there are usually many more per cell. And in the case of a single cell eukaryotes, these organelle is used for locomotion. But in other types of cell, like epithelial cells of trachea, cilia are used for moving particles or fluid over the cell surface. So you can get constant beating of cilia on these tracheal cells to move fluid and particles across. So you can get constant beating of cilia on these tracheal cells to move fluids and particles across them. So on this right-hand side with a time-lapse video microscopy, the image of the flagellum of a sperm cell in action. If you could follow just this one point right here, this one point, you can see that you can get a bending in the neck region, then a wave is propagated through the flagellum to get that whipping motion that can propel the cell in a particular direction. The exonymal dynein, which are the minus end directed microtubule motor proteins, play an important role in the beating of the flagella or the cilia. This is a cross-section of a cilium or flagellum, and the arrangement of microtubules and proteins in the cilium or flagella that is known as the axoneme. Most of them have a same basic structure. There are 9 microtubule doublets that form this circular pattern as shown here. Each of these doublets are made up of A tubule, which has 13 protofilaments, and the B tubule that has 10 protofilaments. And then you have 2 singlet microtubules in the center stabilizing the whole structure. This is often known as the 9+2 arrangement because you have 9 doublets in a circular pattern and 2 singlet microtubules. You can see that the A tubules are interacting with the exonymal dynein, which is what we are focusing on here. So these right here, these are the small branch structures with these globular heads on the end are exonymal dynein, and we will look at these in a little more closely at this structure in a minute. The whole axoneme is held together by the action of certain proteins. So let's talk about these proteins first. So here are the nexin that you find connecting the doublets to each other. So you see the nexin in between the A tubule and the B tubule of the adjacent doublet. You have proteins that make up what is known as the radial spokes that is shown here in light blue. You have the proteins that make up the bridge or the central bridge, and these proteins held the 2 singlets together. And here you have a group of fibrous protein that make up what is known as the inner sheath that is shown here in the light green. And they also stabilize the core of the structure. And here is an EM showing the 9+2 arrangement of an axoneme. So What we are interested in here is the exonymal-dynein. It is an extremely large protein. It has a region known as the base. This region is known as the heads. It is very large, and you can see this actually on an EM image right here. So here is how it interacts with the doublet microtubules in the exonym. You have the base region that forms a very stable association with the A tubule right here. And then you have the head region that forms a transient interaction with the adjacent B tubule. So this is an overall structure of the exonymal dyneins. So this is how it works. Here is the structure of the exonym, and these small proteins shown in the red that are the exonymal dyneins. You have the A tubule on one doublet and the B tubule on the adjacent doublet. You can see that the head region of the exonym is interacting with the B tubule, and the base region of the exonymal dynein is interacting with the A tubule. So what happens is that this is the minus end directed motor protein. So the head region is going to pick up and try to move towards the minus end of the microtubule. But because this structure is so stable, and this is especially for the nexin proteins, the nexin links are shown here in the green, because these are so strong that there is a bending of this flagellum because of the force of the exonymal dynein trying to move towards the minus end. So you have this bending. What happens is the bending is translated into a wave, and then this wave is propagated through the flagellum. So the evidence for this model of exonym function or flagellar function comes from this following experiment. So an experiment was done where the nexin linkers were cleaved by a protease. So now you do not have that strong interaction in between the doublet microtubules. In this case, what happens that when these dyneins move towards the minus end of the adjacent B tubule, you see a sliding of the two microtubules relative to each other, or the microtubule doublets relative to each other. And the flagella don't produce a waveform. This supports the model of exonymal dynein generating the bend in the flagellum that is propagated into a wave. So several years ago, an observation was made in which there was an evidence that there was trafficking of particle in vesicles from the base of the flagellum or the cilium to the tip of the flagellum. The tip is shown here on the top of the slide. Let me orient you that these green lines are the microtubules. The plus end is towards the flagellum tip, and the minus end is towards the flagellum base. What is interesting is that you can see the constant moving particles and vesicles both towards the tip and away from the tip. These are mediated by the kinesin 2 or the cytoplasmic dynein, depending on which direction the particles or vesicles are moving. So when these particles are moving towards the plus end of the microtubule, you have kinesin 2 at work, and when they move towards away from the tip towards the minus end of the microtubule, you have the cytoplasmic dynein at work. This is known as intraflagellar transport, and the role of intraflagellar transport is mostly for maintenance of the flagellar structure. When particles and vesicles are moved from the base of the flagella towards the tip, you are building new flagellum because most of the growth of the flagellum happens at the tip. And when the particles and the vesicles are moving towards the base, it is thought that the receptors on the surface of the flagellum or cilium are being trafficked into the cell. This may be the way down to regulate their function or to take them off the flagellum membrane and traffic them towards the cell. Now, you might be wondering how, if a kinesin molecules deliver its cargo to the plus end, how does it get back towards the base? Or does it get back to the base? There is an evidence that kinesin can hitch a ride on particles that are being driven by the cytoplasmic dynein towards the base. This is also how the cytoplasmic dynein molecules get back towards the tip, as shown here. You can see the kinesin delivering the vesicles, particle towards the plus end, and you can see that there are a couple of cytosolic dyneins hitching a ride. So this is how the cell regulates the trafficking with the flagellum, and this is really important for the maintenance of the structure of the flagellum and regulating the signal transduction within a flagella or cilia. So flagella and cilia, their main functions are for locomotion, or in the case of trachea cells, for moving air, particles, fluid across the epithelium. But in most vertebrate cells, there is also a sensory organelle called a primary cilium. And I'm going to show you in a picture in a minute. You can think of this as the cell's antenna because they play a role really important in sensing what is going on in the environment. Example of well-characterized primary cilia includes those cells in the olfactory bulb, so they play a role really important in sensing odorant molecules. There are sensory organelles in the rods and cones of the eyes, so they play a role in photoreceptions. There are primary cilia on certain kidney cells. It is thought that they can sense the flow of fluid like urine across the epithelium by the angle which they are bent by the flow of the fluid. So these are some examples of functions of primary cilia, but they are not the only ones. Most vertebrate cells have primary cilium. They are extremely stable. They are made of microtubules, and their structure is very similar to the flagellar axoneme that we just talked about, but they are much more stable. So if you treat the cells with colchicine, you will remember that colchicine is one of those drugs that at certain concentration can depolymerize the microtubules. But at even a very high concentration of colchicine, you can retain the structure of the primary cell. So the microtubules within them are much less sensitive to colchicine as compared to the other microtubules within the cell. They are not used for locomotion. They are not motile because they lack the central singlet microtubule, which is so important for the flagellar function. So what I meant by this is here is a cross-section of the primary cilium that you have a microtubule doublet around the edge, but you don't have the central structure, which is important for locomotion. It is the part of the structure that holds the whole thing together. This is a scanning electron micrograph of a kidney epithelial cells, and you can see these primary cilium, some of these cells. And again, in this case of kidney epithelial cells, the primary cilium senses the flow of liquid or urine by sensing the angle by which the liquid flow is bending them. So for a long time, scientists didn't pay much attention to the primary cilium until it was discovered that mutations in certain genes that affect primary cilium structure or function can lead to genetic disorders known as ciliopathies. I'm going to give you two examples of these. The first one, a very common one, is known as an autosomal dominant polycystic kidney disease. It affects about 1 in 1,000 Americans. Symptoms include a cyst formation in kidneys and eventually kidney failure. And it can affect organs other than the kidney as well. You can have an enlarged liver as well as pathologies in the pancreas and blood vessels. The underlying cause of the polycystic kidney disease is a genetic mutation in the genes known as PKD1 or PKD2. So in the majority of the patients, about 86% of them, they have a mutation that renders PKD1 inactive. And for the remainder of the patients, approximately 14%, they have a mutation in PKD2. So PKD1 encodes a protein known as polycystin 1 or PC1, shown here. And the second one, the PKD2, encodes for a protein which is known as a polycystin 2 or PC2. The function of these are not well understood, but it is known that they are localized around the outer membrane of the primary cilium, and they play a role, an important role, in sensing the flow of urine over the kidney epithelium. So in their absence, the patients have a defect in sensing this in a result in the symptoms that are associated with the polycystic kidney disease. The other genetic disorder that I want to mention is the Bardet-Biedl syndrome. It is much rarer than autosomal polycystic kidney disease. It affects about 1 in 140,000 live births in the US. It has an enormous array of symptoms that include obesity, polydactyly, which is extra digits, extra fingers and extra toes, cognitive impairment, kidney, cyst, retinal degeneration, and I put etc. here because the list is symptoms is a mile long. The underlying genetic cause of the Bardet-Biedl syndrome is a mutation in one of the 14 genes that encode the components of a protein complex known as BBsome. You will recall that we were talking about the trafficking from the endoplasmic reticulum to Golgi, or Golgi to the cell surface, or cell surface to lysosome, through the receptor-mediated endocytosis. That when vesicles form, they are often coated by proteins either known as a COPS or the collathrin, depending on the trafficking pathway. Well, as it turns out that the BBsome is a large complex of proteins that interact with the receptors that are destined to be trafficked to the tip of the primary cilium. So here you can see that in red, some kind of sensory receptor that is being trafficked or delivered towards the tip of the primary cilium. You can see the cytosolic portion of the receptor interacting with the protein complex, the BBsome. It is the BBsome that participates in delivering those receptors to the surface of the cilium that can receive the signals. So in the case of a primary cilium, in the olfactory bulb, this would be receptors that would detect odorant molecules. In the case of kidney cells, it would be the PDK1 and the PDK2, which produces the PC1 or the PC2 proteins. In the case of rods and cones in the eye, it might be the receptor that is going to detect light, such as photoreceptors. So you can imagine if you have a mutation in one of these genes that encode the components of the BBsome, and the BBsome is non-functional, what will happen is that you will not get the delivery of these BBsomes to these primary cilia, and these primary cilia will be non-functional. So I think it's clear now that why the Bardet-Biedl syndrome is so severe, because the BBsome functions in so many different tissues. For example, in the kidney, in the eye, in the ear, practically every tissue in the body relies on the BBsome for delivery of specific receptor to the primary cilium. So you can imagine if that is defective in many tissues that are involved, and you can get a wide array of symptoms. What is so surprising is that the effect is so pervasive in all cells in the body, yet it is still compatible with life. It is a very rare ciliopathic autosomal recessive disorder, but once in a while, you get a live birth. We will conclude this chapter appreciating this amazing artwork that summarized this chapter. This illustration is from a review of the ciliopathy, and you can see the membrane of the cilia showing the presence of these PC1 and the PC2 complexes. The illustration shows the microtubule organizing center at the bottom of the cilia. It also shows both the kinesin and the dynein motors and their respective IFT complexes and their cargoes. You can see that there is an anterograde transport by these green kinesin and its blue cargo, and how the dynein is actually hitching a ride with them. On the right side, during the retrograde transport towards the minus end of the microtubule and towards the cell body, the kinesin delivered the cargo already and is now hitchhiking with the dynein, which is now picked up the pink cargo and being delivered towards the cell. This concludes Module 4, Part 3, and also concludes Module 4.